In the mid-2000s, the researchers discovered that a lot of the bacteria consist of this repeated sequence, as shown in these black diamonds here, that flank with the unique sequence as shown in the color boxes in their genome. That can be called the CRISPR, which is an acronym that stands for the clusters of regularly interspaced short palindromic repeats. They found that this unique sequence comes from the DNAs that are found in the virus. And the scientists also identified nearby the CRISPR associated or the Cas gene. These Cas genes were consistently found adjacent to the CRISPR loci and displayed the motifs characteristics of both helicases and the endonucleases. And the tracer RNA sequence was also found nearby the Cas genes. So here is a simplified diagram to show how they are operated. And the CRISPR adaptive immune system is composed of two main phases, the immunizations and the immunity. In the immunization phase, the Cas proteins will form a complex which will recognize and cleave the viral DNA into pieces. And then these viral DNA pieces will then incorporated into this CRISPR loci to form a repeat and a spacer unit. So in immunity phase, following the reinfections, the repeat and spacer units are transcribed to form a CRISPR siRNA precursor. And then these Cas proteins will be guided by the tracer RNA to bind with the siRNA to form a siRNA tracer RNA Cas complex. So this complex is matured after the cleavage by the RNase 3 polymerase. And then so this matured siRNA will serve as a guide RNA that will recognize either the viral DNA or sometimes it could be the viral RNA, and then triggers the cutting ability of the Cas protein to cleave the genetic materials into pieces. So this really is the step where it allows this system could be harnessed as a gene editing technology.